going to do Chapter 6, Microbial Growth. It always happens when I start. So, the requirements for growth. So just so you guys know, you should have an invite, and if you use the invite to get into the presentation, I don't have to keep going over and admitting people. So check for that invite to see if you got it. Okay, here we go. So requirements for growth for microbes. There are the physical requirements and chemical requirements. And the physical requirements include temperature, pH, and osmotic pressure. And what I mean by osmotic pressure will be the concentration of salt or sugar. Sugar would be more if you were in a industrial setting, not an environmental setting. And then the chemical requirements, well, obviously we need carbon. This is a carbon-based world that we live in. Nitrogen is very important, of course, for all of the nucleic acids and amines. Sulfur and phosphorus, you need all of those. Trace elements are very important for enzymes. Oxygen, some microbes don't want oxygen around, so that requirement depends on the type of microbe that you're looking at. And then other organic growth factors that we'll go through here. So first we look at the physical requirements. There'll be a minimum growth temperature where you're just getting a little bit of growth, your optimum growth temperature, and that's where we see in each one of these peaks. It's the middle of the peak. And the maximum growth temperature is how high your particular organism can go. And then we have specific types of organisms. Psychrophiles are cold-loving, so those are the organisms that are going to grow in really extremely cold environments. They're also the type of the psychotrophs are really the ones that get into our food stuff that we freeze or refrigerate. Mesophiles are the moderate temperature loving. Mesophiles are really going to be our entire microbiome. So everything that exists on a human, on animals in nature, those are going to be mesophiles. They really have an optimum growth temperature of 37 degrees Celsius. The thermophiles are heat loving. And so those, of course, are going to be more of our extreme environment type organisms. And then we even have the hyperthermophiles that we find not only in places like Yellowstone Park, but in deep vents of the ocean where you've got these lava vents, very, very hot. So the psychotrophs, I mentioned that they grow in our freezers and our refrigerators. So they really like to grow between 0 degrees, 20 to 30 degrees Celsius is their sort of maximum. And they cause a lot of food spoilage because we tend to leave things in the refrigerator and the freezer that are not sealed really properly. Then when we get up to our thermophiles, we're looking at an optimum growth temperature of 50 to 60 degrees Celsius. Those are found in hot springs and organic compost. So that's not the super hot ones, but the hypothermophiles, those are really the ones that you're going to find in the extreme heat environments of exceeding 80 degrees Celsius. And then, of course, this is a picture of Yellowstone Park, some of those pools, and that's all microbial growth that you're looking at. The blue in the deep pool that's sort of at the bottom of your screen, that's going to be the hottest hypothermophiles there. And then we go out to the thermophiles where you find the orange, and there's a multitude of different organisms actually in here where we can see the orange are probably algae, but they could also be bacteria that are able to capture light, phototrophs, and also archaea. pH. Most bacteria are going to grow in a neutral pH, between 6.5 and 7.5. Molds and yeasts, which we're going to be working with as soon as I can get that I just have to finalize my rubric, and we'll get that all up for you guys this week, and then it's going to take a couple weeks for that experiment. It'll be kind of fun. But they like a little bit more of an acidic environment, and a lot of that has to do with the fermentation process that a number of these go through. Acidophiles grow in acidic environments, very acidic. So here I put up a picture of the Berkeley pit, which is outside of, or not outside of, it's right smack dab in the middle of the city of Butte, Montana. At the surface, between those first 2 inches, we're at a pH of about 2.5, and then it gets lower the deeper you go. And back in the 70s, the 80s, when they really started to, this thing started to form because they stopped pumping the water out. Won't even get into how that all happened, right? They thought nothing could survive. Nothing could live in such an acidic environment. Well, then, of course, microbiologists got involved. We got interested and started testing it. And sure enough, there's a lot of life forms in there. And I actually have a few of those in my freezer. At SKC that we were investigating, but that project is on hold, so it's in the freezer. Now, osmotic pressure, as I mentioned, that's going to be salt or sugar, but sugar would be more in an industrial environment. The whole idea about osmotic pressure is that you're creating a hypertonic environment where you have higher solutes outside of the cell than inside of the cell. And that's going to cause plasmolysis, so a disruption of the cell plasma wall. And it's due to that high osmotic pressure. So water wants to follow solute. We did that a couple lectures ago. So if you have a ton of salt on the outside, you're going to have water leaving the cell is going to die. Now, some extreme or obligate means you're obligated. It has to live in this type of environment. Halophile, halo is for salt, and phile means loving, requires a high osmotic pressure. So these halophiles live in very high salt environments. Facultative halophiles are the type of organism that can tolerate a high osmotic pressure. So that would be a microorganism that you would find maybe in an estuary type environment where you've got fresh water coming in from a river source, and you've also got this ocean or sea, and so you're kind of mixing in that area. But you're going to find these really extreme or obligate halophiles in places like the Dead Sea. So that's the picture we're looking at here, and you can actually see the salt crusted around all of those rocks that are there. And yeah, sure enough, microorganisms do live there. I'd like to go there someday. That would be fun. So chemical requirements. The chemical requirements mostly, well, we'll start with carbon. And carbon, because we are carbon organisms, microorganisms, it's a carbon planet. It's the structural backbone of all organic molecules. Chemoheterotrophs use these organic molecules as energy, so they're going to break down other sugars and things for energy, whereas autotrophs will use gaseous carbon dioxide and fix that into sugars, just as we saw in our metabolism last week. Other chemical requirements include nitrogen. Because nitrogen is a component of amino acids, it's going to be building the proteins, because amino acids are the building blocks of proteins. Nitrogen is found in DNA and also in ATP. Most bacteria will decompose protein material for their nitrogen source. However, there are some bacteria that can actually use these different amines and nitrates from organic material. So these bacteria, we tend to find those deep in the soil, and they're also very useful in degrading our organic material. So when you have a wastewater pool, you want to be able to find some of these bacteria either to dump in there or to wherever you're going to have your wastewater going. You need to get these nitrates and amines out. And the reason you need to get this out of the water is because if you have a really high nitrogen concentration water, That's going to promote growth such as algae, a lot of algae, and algae can clog up waterways, kill fish. It can be a problem, that type of algae. So some of the wastewater that we have in our region, they don't do that here in Polson, but down outside of Pablo, the wastewater, after it's treated from the pools, it's actually shot with a UV to kill anything that might be left over, and then it's sprayed over fields. And so there's a number of fields that farmers down there lease, and they get this water that's high in nitrogen, nitrates, and amines, and it really promotes the growth of the types of whatever they're growing there. I think one of the fields is alfalfa. And I mean, they do have to measure that, because if you have too high of a nitrogen and then you feed that alfalfa to a cow, you can kill it. You really can. So you do have to be careful, but they are measuring what the amount that's going out there, and it has been extremely successful for the past 10 years. They're getting a lot of growth, and then that water percolates down. The nitrogen is taken up by the bacteria that live in symbiosis with that plant life, and then the water, that by the time it hits the water table, it's pure, clean, great drinking water. So we have those few bacteria that use this nitrogen and nitrogen fixation as well, and they are going to be using nitrogen. N2 means gaseous nitrogen, so from the air. There's very few that actually do that. Most of them are going to be getting this from organic material. Sulfur. Sulfur is used in 2 amino acids, methionine and cysteine. So those are the 2 amino acids. And then we also find sulfur in some vitamins, so vitamin Bs, thiamine and biotin. Most bacteria will decompose protein for their sulfur source. Some bacteria, though, can use sulfates or an inorganic H2S, which they find from the environment. We find a lot of these sulfur-loving bacteria in mining environments or old mines, where you had a lot of extra things entered into the environment. And what these bacteria tend to do is they use sulfur as their final electron acceptor, but they're taking up some of these other contaminants, like mercury. And so while they're using sulfur in their electron transport chain, or at the end of their electron transport chain, they are actually taking up some of those bad materials that were left over and sequestering them. Now, exactly how they sequester them is an interesting question, and that is something that we're really starting to look at. There's some specific genes, and it seems like some of them are able to metabolize them into non-harmful substances. And then if you have the MER-A and MER-B genes both turned on for mercury, they're going to degrade it. But if you only have one of those genes, then basically you're taking this up and keeping it as basically a little poison pack. So it's interesting. Phosphorus. Phosphorus is the DNA backbone, right? You've got the big phosphorus there holding onto the groups. RNA, of course, and also ATP. We've seen the triphosphate, right? There's 3 of them. Also, phosphorus is found in membranes, so we have the phospholipid heads. PO4 is usually the source of phosphorus, and that's a different cycle. When we get to the environmental chapter, we're going to look again at those different cycles, including the phosphorus cycle. Trace elements. Trace elements include inorganic elements. These are required in very small amounts, and they're usually as enzyme cofactors. So these can include iron, copper, molybdenum, and zinc. I didn't put on here some of the others that we use in the lab, but basically a lot of enzymes are going to require some kind of cofactor. So if you are ever working in a lab and you're working with microbes and trying to get them to grow, you're always going to want to make sure that you put in some of these trace metals for them. So oxygen. Some are going to require oxygen, some are not. Our obligate aerobes require oxygen. They absolutely have to have oxygen to survive. Facultative anaerobes, these can grow with fermentation or anaerobic respiration when oxygen is not available. So this means that they can survive, or they can use oxygen. They like to have oxygen around because it gives them lots of energy. But if oxygen isn't available, they can go down another pathway and use fermentation instead. Anaerobes are unable to use oxygen, and most are harmed by it. So if this is a strict anaerobe, it's going to get away from any oxygen source. They don't have the enzymes to break down oxygen radicals. Most anaerobes are going to be found in places where you don't, they'll be deep in tissue if it's a pathogenic one. That's where you can get the really black necrosis. That'll be a pathogenic microbe that's anaerobic and it's deep in the tissue. Some anaerobes, we have some anaerobes in our teeth. And so when you've got a rotten tooth, it's deep in there, that's going to be an anaerobe. The bottom of the lake, we have a lot of anaerobes. Most ponds and such like that have anaerobes at the bottom, and you can smell them. So pond flipping is when we have a change in the environment. So twice a year really is when this happens, although it's Montana. So when things go from cold to hot really fast, or hot to cold very fast, you're going to change the pressures in your water systems, and your pond essentially flips. So everything that's on the bottom kind of gets disrupted up to the top, and then they float back down. But for a day, you're going to probably smell a really bad smell. And so if you've ever gone to the dog park and you smell that, it's not actually the ponds, and it's not what's being degraded in the waste products. What you're smelling are the microbes. You're smelling the methanogens. It's one of the things I do when we go out sampling and we're pulling our dirt from deep in the lake, well, pretty deep. We're pulling up anaerobes, and I'm always like, do you smell the methanogens? And everybody looks at me like I'm crazy, but you can tell if you've got an anaerobe in there. Aerotolerant anaerobes, they can tolerate, but they can't use oxygen. So they've got the enzymes to deal with oxygen radicals, but they're not going to use it for any energy source. And then we have the microaerophiles, and they require a very small oxygen concentration, which is lower than the air. Every time I start lecturing, I start dying of something. So basically, a microaerophile can't handle a very a normal concentration, that 21% concentration of oxygen. And so you might find these microaerophiles in deeper water sources or on really high mountaintops where you've got a lot less pressure of oxygen. Okay, so we talked a little bit about those oxygen radicals, and so this is why some of these anaerobes that can't use oxygen, they can be harmed by this. So singlet oxygen can be boosted to a higher energy state, and it becomes reactive. We get these superoxide radicals because most sources of energy are going to have oxygen and water around them, right? So when we looked at all of our metabolism, we had oxygen as the final electron acceptor, and we have water as a byproduct. So anything that's going to be using oxygen has this potential of the oxygen and the water reacting. And when they react together, you can end up creating hydrogen peroxide. So we don't really need to go through all the chemistry here, but H2O2 is hydrogen peroxide. It's a radical that's produced. It can react with oxygen to create this hydroxyl radical there at the bottom, or it can be degraded, and it's usually degraded very quickly by catalase. So I will have you all doing a little bit of that test when you grow your own fungi. You'll just take a little toothpick, take a little bit of that fungi, put it on a paper plate, and put a couple drops of hydrogen peroxide. You'll know if you have catalase in there because it'll bubble. If it doesn't bubble, then you don't have readily available catalase. And we might not see much bubbling actually because a lot of molds are facultative. And so that means that they can go the fermentation route. So they might not do this, but it'll be fun to just find out what you grow. All right, so for the organic growth factors, organic compounds can be obtained from the environment, and these are going to include vitamins, any essential amino acids, meaning amino acids that can't be made, pyrimidines and purines. Most microbes have that pathway. They have the deuteroph-enteroph pathway, and they can do that because they have pentose. They're able to make pentoses, but some microbes can't, so they're just going to get it from the environment. All right, on to biofilms. So biofilms are very useful because they're microbial communities, and they can form a slime or a hydrogel. They can adhere to surfaces, and they can communicate with each other. And they use their communication that they're using is called quorum sensing. So they don't actually, call each other up on the phone. They can't actually see, but they can use different chemicals to sense where they are. So bacteria will secrete an inducer, which is a signaling chemical, to attract other bacterial cells, also known as chemotaxis, if you remember. We did use that word once before, and we will be using it in the immune chapters. So the benefits of a microbial community is that they can share nutrients and also shelter each other from harmful environmental factors. Biofilms are often found in the digestive system. So you're going to find a big biofilm down in your gut. We find biofilms formed in sewage treatment plants. They are a good and a bad thing. We do need the microbes in there, but the bad thing is if they grow too big, they'll clog up the pipes. So you kind of have to flush them out now and then. They become much more resistant to microbicides. So anything that's going to kill microbes, it's going to be harder to kill them when they're in a biofilm formation. We also find biofilms in a lot of different infections. And so anytime you put anything into the body that's not naturally supposed to be there, so any kind of mechanical heart valve, catheters, anything that's plastic or has a different material, if that's put into the body and it's done in a non-sterile manner, if it's exposed at any point to bacteria, you can develop a biofilm. We find this is actually pretty serious with catheters. So if somebody has an extended catheterization, almost inevitably they will get an infection. So, it's recommended to try to minimize how long somebody is catheterized if possible. So culture media, how we're going to grow things. In a culture medium, we need to have nutrients that are prepared for microbial growth. We want to make sure that this media is sterile, that we don't have anything already alive in it, because we want to introduce our inoculum. We want to introduce the microbes of our choice into a medium. And then we grow it, that's culturing. The microbes will grow in or on the culture medium. And there are different types of culture media. We can use a solid culture media, agar. Agar is a complex polysaccharide, and it's actually used as a solidifying agent for culture media. And this is found in Petri plates, so that's what you're looking at there. Those are plates. Slants, That would be a little glass tube with the auger at a slant. And deeps would be a glass tube with the auger in the equally on the bottom that you just stab. Agar's generally not metabolized by microbes, and that's why it's a very good solidifying agent, because we can introduce into the agar any kind of media growth that we want. So whether we want it to be selective, differential, or nutrient only, we can put that into the agar, and then we can select what kind of microbes. And so we're going to look at those different types in a moment. Agar liquefies at 100 degrees Celsius, so we can sterilize it. We use an autoclave, and we get it into a liquid form, and then we pour our plates, and it'll solidify just above room temperature, so it solidifies pretty quickly. So there's a couple of different kinds of culture media. Well, there's many different. But they can be divided into groups. Chemically defined media is when the exact chemical composition is known. Fastidious organisms are those that require many growth factors provided in chemically defined media. So if you've got a particular microbe that you want to grow and you know exactly what it needs, this is the way that you would go. Here are some examples. So the take-home message here is that you're going to see in grams, liters, milliliters, the exact amount of whatever constituent it is put into this media. So we have the glucose at 5 grams, specifically 5 grams of glucose. I'm not going to read all of them, but you get the idea that you need those exact measurements. Now, complex media, in complex media, you're going to have extracts and digests of yeasts, meat, or plants, and the chemical composition will vary batch to batch. Now, when we look at the example here, they've got peptone, beef extract, sodium chloride, agar, and water. Now, sodium chloride is a chemical, NaCl. So 8 grams of NaCl is known. We know the molecular weight of sodium chloride. We know the molecular weight of agar. That's not going to change. And we know what water is as well. So those sodium chloride, agar, and water are chemicals, but because we're combining them with 5 grams of peptone and 3 grams of beef extract, that whole composition right there, that's a complex media. Because partially digested protein and beef extract, even though I'm measuring them out in grams, I don't know every single chemical that's in there. So I noticed a lot of people did this last quarter when I was teaching microbiology. They were taking the components and they were saying which was complex and which was chemical. That's not what we're looking for. You have to look at the entire table. So that entire table at the bottom there, that's a complex media. It's not complex and chemical. The whole thing is complex because we have peptone and beef extract in there. The top 2 tables, those are chemically defined. All of them are chemically defined. We find our complex media in nutrient broth and nutrient agar. So this is usually where you're just trying to get some growth and you're not trying to separate anything out. All right, so specific growth media for anaerobes. We use a reducing media. It's for the cultivation of anaerobic bacteria. It will contain chemicals such as sodium thioglycolate that combine with oxygen to deplete it. And then there has to be some kind of heat source to drive off oxygen. So sort of depends on what kind of tools you have. Now, if you have a chamber, an anaerobic chamber, that's going to already create an environment that is anaerobic in that big chamber. And you have to use those gloves to get in there. I don't know why that person has bare hands. They should be wearing gloves. But if you don't have an anaerobic chamber, they're very expensive and very expensive to maintain. You can use this little anaerobic jar. And what it has in there is it has a little catalyst. It clamps down. It has a little palladium catalyst. You have a packet that you open up, and it releases CO2. And then what you've got is it pulls out the oxygen in there, that catalyst does. And then you usually have a little indicator in there so you know when your little chamber has turned anaerobic. And then you have your plates in there. Anaerobes grow very slowly. It takes a long time to get them to grow. Some other types of anaerobes are the capnophiles. And these are microbes that require a high CO2 condition. So you can use a CO2 packet and or a candle jar. We don't really use the candle jars so much anymore because we can just add a little packet in there that'll take up the oxygen, and then you add another packet that's going to release some CO2. And that's the kind of, it's just easier. But this is kind of the old-fashioned way, and we still kind of call them candle jars, but we don't actually have a candle in there because that's a fire hazard. All right, some other special culture techniques. This is biosafety levels. Often biosafety levels are not for the human that's culturing. Often it's for what's being cultured. You need to keep that organism safe from you because we're covered in microorganisms. And that's really the case for BSL 1 through a lot of time through 3. Like in our labs at SKC, we don't have anything that's going to hurt you. We are just trying to protect the organisms, not you. But BSL 1 is a basic teaching lab where you just have some ethanol that you spray and you wear gloves. BSL 2, you might pop on a lab coat as well and some eye protection. BSL 3, you're going to put everything into a cabinet to prevent airborne transmission. Again, when we're at SKC and we're doing stuff in the hood, that's what we call it, is the hood. It's because I want to prevent airborne transmission from you to whatever our experiment is. But in some cases, in some labs, like down in the Hamilton labs down here outside of Missoula, they do actually work with some airborne microorganisms. You can also argue that you don't necessarily always want to work with fungi, a lot of fungi, because they can become airborne. I mean, we're going to be growing a little bit on bread. We're not going to be doing anything that you don't clean out of your refrigerator on a regular basis. But if you are working with really culturing a lot of these, you might want a fungi room. And you know, every time that you would open your plate or whatever it is, you do that in the hood so that you don't get spores. Now, BSL 4, you have to be in basically this little space suit. So this is actually Marshall Bloom, Dr. Marshall Bloom. He works down at the Hamilton Labs. He comes up to SKC on a regular basis, and we go down to visit pre-COVID, of course. Hopefully we'll be able to do that again. But they have a BSL 4 lab. And this is where the exhaust air is filtered twice through HEPA filters. You are not exposed. You have your own environment. And then you also work in a hood. And it's not comfortable. I mean, if you can see, he's in a spacesuit, right? Trying to work with very small things in a hood with a spacesuit on. Research goes pretty slowly. So that's why we always try to come up with other techniques, such as virus-like particles instead of a whole virus, so that we can more readily move research forward without BSL 4. All right, so the different types of media that we have here are differential media. Differential media will allow distinguishing of colonies of different microbes on the same plate. Some media have selective and differential characteristics. Oops, my words got written over a little bit down there, but we have McConkey agar, which is a differential media, and we're growing a couple of different organisms here. We have Staph epidermis and Staph aureus, and you can see just by culturing them how quickly you just glance at it and go, these are two different microorganisms. The aureus actually is changing the pH of the McConkey agar, and that's why it's turning yellow. Both can grow on this plate, but only one is going to give that really distinctive characteristic. Blood agar is another differential medium where you can have different microorganisms grow, but some are going to lyse the red blood cells, and then they give a halo around them. And then there's different types of the lysis. We have alpha lysis, beta lysis, and then there's no lysis at all. And so you can grow many different organisms on there, and then just look for the ones that are lysing it. An enrichment culture will encourage the growth of desired microbes by increasing very small numbers of a desired organism to detectable levels. This is usually a liquid. And so most of these will be just a liquid media like we did. Actually, we did this last quarter. We had a few people come in to do lab. They had soil samples, and we had an enrichment culture phenol liquid that encouraged the growth of soil microbes. And it did that because you have very few of them, very few went in there, and they just grew and grew and grew until we had some very cloudy, nice cultures. All right, so to obtain pure cultures, a pure culture contains only one species or strain. And a colony is a population of cells arising from a single cell or spore, or a group of attached cells. I really want you to have the take-home message of a single cell. A colony is often known as a CFU, or a colony forming unit. And we use the streak plate method to isolate pure cultures. The streak plate method, okay, they have 4 here. I usually only use 3. You streak your inoculum only in quadrant 1. So just one streak there. Then you have to get another loop or sterilize your loop depending on what kind of tool you're using. In streak 2, you go back. You can see the line goes back into the inoculum a couple of times and crosses over. 3 crosses into 2, 4 is crossing into 3. The reason that we have just a few crossing over, and actually they have a lot, I wouldn't cross over that many times, because when you see the colonies that grow, they're pretty thick. And you want isolated single colonies, beautiful isolated single colonies. You don't want the colonies to touch each other, because then you're having a mixing of cells or mixing of strains. And what this whole process is supposed to be doing is it is supposed to be diluting your original inoculum. So every time you streak out, you're just pulling those cells further away from each other so that they have enough space that one cell will develop into a colony without touching any other cell. How we preserve these bacterial cultures. One of the ways would be deep freezing or lyophilizing, which is just freeze drying. So you can dehydrate your bacterium in a vacuum and then bring them back to life. They don't actually have this on here. This method of preserving bacterial cultures is going to kill a lot of your bacteria. So really the way that, I And I don't know why they don't have this in the book, but it's probably because these people have been out of the lab for a long time. The way that we generally are going to preserve our bacterial cultures for long periods of time, for years, is you're going to take your colony that you got, and you're going to put it into a little liquid culture, so maybe a really small tube. You're going to let it grow up a little bit in there, maybe 4 hours, because that's enough divisions. And then you're going to add glycerol. And then you have this little small packet, basically, of a bunch of cells, and really it's millions of cells. And the glycerol wraps around the cells and it protects them from freezing. You put those in at -80, and then they're fine. They say after 2 years you should reculture and get a new freezer, but I've been able to revive things after 4 years, 5 years. Okay, so let's specifically look at the growth of bacterial cultures. So why we're saying that a single cell can give us this very pure colony of millions of cells is because of the way that bacteria divide. They increase in the number of cells, not the cell size. So when you initially streak out a plate, you can't see anything on that plate. Then you start to see your colonies develop. And that's not because the cell got bigger, it's because it divided again and again and again and again to make many, many cells piled on top of each other. Bacteria divide by binary fission for the most part. So one cell becomes two, two cells become four, so on and so forth. Budding is more for fungal cells, kinetospores, and the fragmentation of filament will also be with fungi. Now, when we get into the chapter that just talks about fungi, we're going to talk more about that division there. But right now, I really want you to concentrate on the binary fission.
Bacterial cells do not divide by meiosis and mitosis. Instead, most bacteria grow asexually by a process known as binary fission. In this process, the bacterium copies its chromosome, elongates, and splits off into two daughter bacteria with a genetic makeup identical to the parental bacterium. Each daughter cell can continue the doubling process. This process happens rapidly, so the number of bacteria present in a solution can increase very quickly.
Okay. And because it goes so quickly, we call that quick division the generation time, which is the time required for a cell to divide. And this can be anywhere from 20 minutes to 24 hours. So usually when you start working with a culture of cells and you really need to know your concentrations, you get a feel for it, let's just say. But really, you want to measure your concentration of cells until you are very comfortable knowing how fast they divide. Because if you get a really high cell division and you put things into an incubator, say at 1 mil for an hour, you're probably going to overgrow your cells. You're going to have way too many. You're going to get into that logarithmic and beyond. So binary fission doubles the number of cells each generation. So you get your total number of cells equals 2 to the number of generations. So that's a logarithmic. Growth curves are represented logarithmically. So we can show you kind of these different generation numbers, how they're doing it. And then you can see the exponential growth there on the final picture where it just takes off. Now this is important to know because if you are working with cells, very often you want to have them in logarithmic growth. You don't want to have them at the peak and you don't want to have them at the bottom. You want them actively in the middle of their logarithmic growth. So you really have to time your cell growth and learn how they grow in order to get the perfect amount of growth for your experiment, whatever you're going to be doing. And very often that's going to be inducing a gene or protein production, that sort of thing. So when we look at oh shoot, I guess it's on the next slide. The phases of growth. We have a lag phase, the log phase, stationary phase, and the death phase. So there's always a lag phase, and basically that lag phase at the beginning, so when you're looking at the bottom there, that is where the cells are sort of gathering the nutrients together to start to grow. And then all of a sudden, you hit your log phase. And really, it's fast. And it can be a small amount of time, really, where you're in this log phase. So you might have a lag phase for, say, an hour. And then you all of a sudden hit your log phase, but you only have 20 minutes of log phase growth before you hit that stationary and death phase. So, you really have to time these. And the log phase is active growing. Well, you get to the stationaries when you've used up all of the nutrients, and then the cells start to die, and as they're lysing, they're releasing enzymes and all kinds of toxins and stuff into the media, which will screw up your experiment. So let's go back to this guy for a second.
Most bacteria reproduce by a process called binary fission. During binary fission, the parental cell divides to form two daughter cells. In the first step of binary fission, the bacterium replicates or copies its chromosome and attaches each identical copy to a separate location on the cell membrane. Unlike eukaryotic cells, bacteria do not need to dissolve a nuclear membrane or assemble a mitotic spindle, making binary fission very quick and efficient. Second, the cell elongates. Since the chromosomes are attached to different locations on the cell membrane, they separate from one another. Third, the bacterium pinches off at the center and forms a septum, a separating wall between the two compartments of the cell. Finally, the cell splits into two new daughter cells. These daughter cells are fully mature bacteria that can grow and, if conditions are right, continue the binary fission process.
All right, so moved on to the lag phase, log phase, stationary, and death. So we'll let this guy kind of talk to you for a second. One thing he didn't say in that previous video is those daughter cells are identical. And that's why it's really important to have this binary fission and those single colonies that are not touching, because the genes that could be in this one particular colony may be different than the ones in the other one, all because they arose from single cell. You've got identical millions of cells there. Well, if you're carrying out an experiment that relies on having that exact gene, you don't want your colonies to be mixing, because then you're going to have to separate everything out again and re-isolate your very particular gene that you're looking for. So we'll let this guy go.
Growth in bacteria is defined as an increase in the number of bacterial cells within a population. Growth trends within a bacterial population can be illustrated using a bacterial growth curve, such as this one. Growth occurs in 4 phases: lag, log, stationary, and death. Let's look at each phase of the curve to see what's happening. When dormant bacteria are transferred to a fresh medium with plenty of nutrients, they spend some time switching their metabolic machinery from the dormant state to an actively growing state. During the lag phase, the number of new bacterial cells is equivalent to the number of dying cells, resulting in a horizontal plot on the growth curve. The lag phase can last from less than an hour to days, depending on the species of bacteria. During the log phase, or exponential growth phase, the bacteria are actively undergoing binary fission. During the log phase, the bacteria double their numbers every generation period. The generation period can range from 20 minutes to days, depending on the species. As long as there are plenty of nutrients and little waste buildup, the bacteria will continue to grow exponentially. It's convenient to represent the growth of the bacteria on a logarithmic graph, because the numbers of bacteria increase very rapidly with each subsequent generation. When the log of the number of cells is plotted versus the generation, the graph is linear. After the bacteria have grown for a while and they're starting to deplete the nutrients in the growth medium, some of the cells begin to die. The stationary phase is reached when the number of new cells equals the number of cells that are dying. After the bacteria have depleted most of their nutrients and their waste buildup is getting too great, the bacteria begin to die in larger numbers than are made. If the death phase is carried on for a long time, most of the cells will die. If a cell is to survive, it must switch its metabolism back to a dormant state because there are not enough nutrients available for any more growth.
Okay, so the bacterial cultures, how we're going to measure this microbial growth. So there's a number of different ways to do this. Generally speaking, if you're going to be using the looking for a log phase, you're not going to use any of these at all. But it's important to know that they exist. Direct measurements, you're going to use a plate count or filtration or most probable number or direct microscopic count. You'll be using any of these if you are trying to detect something in, say, a water source. So you want to know if you've got coliforms and how much are there. You might use a plate count. So plate counts are counting the colonies on plates that have 30 to 300 colonies. And to ensure the right number of colonies, the original inoculum will be diluted using a serial dilution. And so that's what we see in the bottom on the left-hand side. You've got your original liquid culture, and then so you take a little bit out and you plate it, and then you dilute it, and then you plate that, dilute it again, plate. And then you can see by the end you're getting down to, you've got it looks like three total colonies on there, but you've got a dilution of 1 to 100,000. So what you can say is 3 cells went onto that plate. How much was originally there in my original inoculum? You're going to have to multiply that times your dilution. So 3 times the 100,000, right? So you had 300,000. Now don't be confused, because if somebody's asking, particularly on the question, somebody asks you how much or how many cells were plated, you would say 3. Somebody says how many cells were in the original inoculum, then it would be that 3 times 100,000. I hope that makes sense. So you want to look at those questions and how they're worded very carefully when you're doing your homework. So then counts are performed on bacteria mixed into a dish with agar or spread under the surface of the plate. That's just very simply how many colonies have grown. Now filtration, this is a particular way to find out how many colonies are in any kind of liquid culture. Again, like a water source where you're trying to see, is this drinkable water? So the solution will be passed through a filter. The filter will have pores in it, so most things go through, but bacteria are very small, right? So they're going to be trapped on the filter. Then you take that filter paper and you plop it onto your agar, press it down, pull it off, put the agar plate into the incubator, and then you'll grow colonies. And then you count those colonies and you say, this is how many cells that I had in my original, or in my water source. And then depending on what is an acceptable level of that bacteria, you can say whether that water source is clean or not. The most probable number is going to use, and this is a liquid culture, and you're going to count how many tubes grow your inoculum. And so you'll just count your positive tubes and you'll compare them with a chart. And so you'll make a statistical analysis of how many positives you get. And this is just, it's a way to do basically what we were talking about with the plate counts in liquid form. Another way to know, so this is direct, how many bacteria are in a source, is a direct microscopic count. So you take your bacterial suspension and you put it on a particular slide. It's called a Peterhoff-Hauser cell counter. And it's on a slide. It's very small, and it has a little, well, it's just as cross hatches. And then you look down and you have a little, it looks almost like a stopwatch. But every time you push it, it puts a number on there, and you just get a number in each little quadrant, and then you do your math to find how many bacteria are there. This is not used anymore, just to throw that out there. I mean, you could, but most people are not going to use this method because we have cell counters, and they're digital cell counters. They also have a nice visual computer screen, and so you can add a quick stain, you can put it into your cell counter. It'll count how many cells are alive, how many cells are dead, and it'll give you a picture. You really don't need to be doing this by hand anymore. And also, when you do it digitally, you're getting your data right there. And that data can go into your computer and be pulled up at any other time. So instead of somebody relying on the scientist to say, that's how many I counted on my little stopwatch thing, they can pull up on July 1st of 2017, this was the cell count, and then you can verify data. So direct microscope count, I would say cell counter is really how it's done now. Now this is still done. Now this is probably the most popular, quick way to measure bacterial numbers, and it's a turbidity measurement. And what it's going to do is it's going to measure the cloudiness, and it uses what's called a spectrophotometer. And this is really the fastest way to find out if you've gone from lag phase to log phase. These are turbidity. So if you've got everything in your liquid culture, it's shaking around in the incubator, you pull out 1 ml and you put it into your spectrophotometer, every cell is going to bounce back the light. Now they're showing this big light bulb. You don't use a light bulb, it's a UV light source. It's very small. And it goes through, and if it's refracted back, you're going to get higher numbers. So you'll get a 0 if it goes all the way through. If it's refracted back, you get a high number. And usually when you're looking for a log phase, you're going to be looking at 0.4 to 0.8 units, and that's the number that you would use. So that's the most common method. It's quick, it's indirect, but it's pretty accurate. And so we can measure our metabolic activity. So we know how many of those cells are in log phase. You can also use dry weight bacteria for filamentous organisms. So you can dry them and then just weigh them on a scale. And that's going to be for fungi. Okay, so we're at the end here. In summary, we have growth requirements for microbes that include temperature, pH, osmotic pressure, and chemicals, and the oxygen or the lack of. Biofilms exist in microbial communities. They aid in microbial growth. Culture media is used to detect our different microbes. We have nutrient just to get everything going. Differential can grow everything on the same place. Selective will just grow one type of microorganism. Culture media can be solid or liquid. Solid is easier to isolate colonies, to isolate your gene from that specific one cell that grew into a colony. Liquid is useful though if you want to, then you took that very specific colony, now I want to express the gene and maybe I want to make insulin, the protein for humans, in a big industrial setting. You've got your one colony, you inoculate it into liquid, and you get to that nice log phase, and then you stimulate your gene to turn on and make your insulin. Colonies are pure cultures that arise from a single cell, and they can be counted. And we can count them using direct or indirect methods. Okay, that is the end. And I wrote end. So I'm going to go ahead, stop our recording. Okay, there it goes.